Okay, we're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at mitosis. Okay, and we're going to start off by looking at the cell cycle. We're then going to move on to the stages of mitosis. And then we're going to be also looking at the mitotic index. And then we're going to finish off by looking at three exam questions, which I've picked up from past papers. If you have any questions at any point, you can let me know in the Q&A. If you don't have access to the Q&A, you can scan the QR code, which is on the screen, um, and ask any questions. But we're going to make a start now. So we're going to be following these points on the specification. This is all about the cell cycle and mitosis and the ways that eukaryotic cells divide. We're not going to be covering the required practical of mitosis in today's lesson, but we will be covering that in a future lesson. So, if we make a start now, we're firstly going to start off with some key points that you will already know, mostly from GCSE. Okay, and that's how eukaryotic cells divide, how prokaryotic cells divide, and how viruses divide. So first of all, we've got eukaryotic cells, and eukaryotic cells, they have two ways of dividing. They can either divide by mitosis or they can divide by meiosis. Those are the two ways in which eukaryotic cells can divide. In today's lesson, we're just going to be focusing on mitosis, but later on in A-level biology, there is also meiosis, which pops up, and we will be looking at the process of meiosis as well. But today, for normal body cells, the ones which have 23 pairs of chromosomes, we have mitosis, which occurs in order for cell replication to occur. Meiosis only occurs in gametes, which have half the number of chromosomes. Prokaryotic cells, again, that's another topic which we will come on to as well. They replicate by binary fission. Okay, so they go through a different process called binary fission for cell replication. And then viruses as well. So we looked at in our first lesson on cells, we looked at eukaryotic cell structure, prokaryotic cell structure, and the structure of viruses. In viruses, they are very small. They are not really classified as a cell because they are non-living and they don't have any internal sort of organelles. They replicate inside host cells. So when we have our normal body cells, when a virus comes into our body, the viruses will enter the host cell, our normal body cell, by a receptor. It will bind to the receptor, and then the normal body cell will take the virus in. Once it's into our normal body cell, it will then replicate rapidly, and then it will cause cell damage. And that cell damage is what makes us sort of feel ill. Okay, so they replicate by injecting the nucleic acid in the cell in order to replicate the virus particles. So as I mentioned, the virus binds to a receptor, which is on our cells. Every cell, every body cell has that specific receptor which the virus binds onto. The virus then binds onto it and then enters the normal body cell, and then it will undergo replication by injecting its nucleic acid and accumulating it with the DNA of the normal body cell. And when the virus particles have replicated too much, like there are too many virus particles present within the cell literally explodes, and that causes cell damage. All the virus particles are released into the bloodstream, and then they will go on and bind to different cells, and that will cause a knock-on effect if it's not counteracted immediately. So those are the three processes um, that we have. So we have eukaryotic cells, we have mitosis or meiosis, we have prokaryotic cells, binary fission, we have viruses, which enter cells via receptor. They replicate by injecting their nucleic acid, and then they have too many virus particles within a cell, which cause the cell to basically explode, releasing all those virus particles, and then that's what makes you feel ill. Okay? So if we move on to looking at the cell cycle, Okay, now there are three key stages of the cell cycle. Okay, and you probably would have looked at mitosis at GCSE, but we're just building on the knowledge from GCSE when you're in year 12 or year 13. There are three main stages of the cell cycle. You've got interphase, you've got nuclear division, and then you've got cytokinesis. Okay, and the main aspect where mitosis occurs is the second part. This nuclear division part is where mitosis occurs. You've got interphase, and then you've got cytokinesis at the end. Cytokinesis is really simple. We will cover it by looking at a diagram, but cytokinesis is basically a fancy word for something you've already looked at GCSE. So basically, when the cytoplasm and the cell membrane divide to form two identical daughter cells, that final process is called cytokinesis. It's the division of the cell membrane and the cytoplasm. Mitosis has a couple of more stages between it, and there's an acronym that you can remember to remember the stages of mitosis. And then you've got a stage at the start, which is called interphase. Again, we will look at that in more detail on the next slide. But these are the three main stages of the cell cycle. You've got interphase, mitosis, and then cytokinesis. Okay, and then mitosis is basically split into four different stages as well. There are four different stages in mitosis. So basically, altogether, you have six stages. You've got your interphase, you've got your four stages of mitosis, and then you've got your cytokinesis. Okay, and cytokinesis you already know, just a fancy word that you just need to learn now. It's just the division of the cytoplasm and the cell membrane to create new identical daughter cells. So we're going to start off by looking at interphase. Okay, and this is in a bit more detail than you would be required to know. But again, it helps for the understanding of interface. And in order to know what you actually need to know, I've picked up some exam questions, which will help you sort of pick up mock scheme answers. So there are three stages of interphase. You've got the G1 phase, you've got the S phase, and then you've got the G2 phase. Those are the three stages. Now, you don't really need to explicitly know those stages like G1, S, and G2. However, you do need to know what the idea of interphase is, what happens during interphase. And I'm going to underline the key aspects that you need to know. In interphase, the organelles double and the cell grows in size. This is the basic sort of main idea that you need to know for A level knowledge. The organelles which are present within the cell, they will double, the cell will grow in size, your DNA will replicate. So again, this comes partly under organelles replicate, and it just prepares for mitosis. Okay, so the key aspects that I've underlined, all you need to know is that when a cell is in preparation to undergo mitosis, in other words, it's going to divide into two other cells, it needs to prepare for that process to occur. And in order to prepare for that process to occur, the internal organelles, you've got your mitochondria, you've got your nucleus, you've got your ribosomes, you've got your rough endoplasmic reticulum, you've got your smooth endoplasmic reticulum, you've got loads of organelles within that cell. All those organelles will need to double and they will need to, and the cell itself, in order to hold those organelles, will need to grow in size. And that's how we know that the cell is ready for mitosis to occur. So the organelles will double in size, the cell will grow in size, and the DNA will replicate. Okay. Now, the second part is not really something you need to explicitly know for A level. However, it's useful to know. So we know that we're preparing for mitosis, and one of the key stages that is also present is the error checking stage. Sometimes when your DNA is replicating, you can have mutations, and if you have mutated DNA, then that can lead on to a knock-on effect. So, for example, if you have a mutated piece of DNA, that DNA will be passed on cell to cell to cell, and it will produce loads of body cells which also have mutated DNA. If you have mutated DNA, you can't code for the normal proteins that you would expect to be produced within your body. And if you are not producing the normal proteins, your body won't function properly. So it's really important that before mitosis occurs, you check if there are any mutations within the DNA. If the DNA has replicated correctly, or has there been some errors between the replication of DNA? You need to check whether the DNA is still intact, if it's still providing the function that we need, and if it still has the sequence of amino acids that we want it to have. So a key aspect of your interface is your error checking stage as well. Your error checking stage will make sure that the DNA replicated doesn't have any mutations. It will check for any potential mutations within the DNA which has replicated. And if there is a mutation within the replicated DNA, the cell is destroyed at that moment to prevent any harm. It will be self-destroyed, and that process is called apoptosis. Okay, so if there is mutated DNA, if there is DNA that is not meant to be there. It's not following the normal base sequence that we need. Then that cell will be flagged and it will be destroyed in that same moment. Because if it is not destroyed, that mutant DNA will replicate, it will be passed on to further cells, it will continue replicating, it will pass on to further cells, and all of a sudden you have so many cells with the wrong DNA in your body. And if you have the wrong DNA, you're going to produce the wrong proteins. If you produce the wrong proteins, you're going to produce the wrong enzymes. You can't catalyze any reactions within your body. And if that happens, then your body will stop functioning as it should be. Okay, so that's a really important stage within interphase as well. But over here, the key idea of interphase, your organelles double, your cell grows in size, and your DNA replicates, and basically the cell just prepares to undergo mitosis. That's what happens in interphase. It's your preparation for mitosis to occur. Now what we're going to do is we're going to move on to the 4 key stages of mitosis. Okay, so remember I said the first stage, we've got interphase, then we've got mitosis, which has 4 stages, and then we end off with cytokinesis. Okay, and the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. And the way that you can remember this is by remembering this acronym, PMAT. If you remember that acronym, you know that P stands for prophase, you know that M stands for metaphase, you know that A stands for anaphase, and you know that T stands for telophase. So those are the 4 stages of mitosis. The way that you remember it is by learning this PMAT. That's the acronym that I learned, and it stands for each of the stages of mitosis. So here's a diagram. We're constantly going to be referring to this diagram throughout the lesson, but at the moment we have just looked at interphase. So if we look at interphase, all we need to focus on is that in interphase, we've highlighted the nucleus, we've highlighted the chromosomes, and we've highlighted the cell membrane. All we need to know is that this cell has sort of replicated its organelles. Okay, so during interphase, your organelles have replicated and the cell has grown in size. That's basically what's happened in interphase, and it's basically decided whether it's going to carry out mitosis or not. And mitosis will only be carried out if it's past that error checking stage to make sure that the DNA has replicated properly. Okay, so moving on to the first stage of mitosis. Okay, now the first stage of mitosis is prophase. Remember PMAT. P is first, prophase is first. Now on this slide, there is a bit more information that you need than you need, but I will again underline the key parts which you need. Okay, a lot of this is just to help you understand what's actually happening in prophase. Okay, so first of all, before mitosis, the chromosomes are invisible. That's what you need to understand. Okay, so before mitosis, the chromosomes are invisible. You can't really see the chromosomes before mitosis occurs. Now, this is the first stage of mitosis. Prophase is the first stage of mitosis. And in the first stage of mitosis, the chromosomes, they condense and become visible. Okay, this is a really important point. Okay, normally in exams, when they ask you to what actually occurs in prophase, this one mark and this is another mark. Okay, so those are two marks in an exam. Chromosomes condense and become visible. Okay. Now, before we have mitosis, before prophase, this is something that you don't really need to know. I've just added some extra information there. Chromatin, which is a mixture of DNA and proteins, condenses to form chromosomes. So the reason why you can't see the chromosomes before mitosis occurs is because the chromosomes are in liquid format. They're in a format known as chromatin. Okay. And when mitosis has been initiated, that chromatin liquid, which was originally present in the cell, that will condense to form these chromosomes. Okay, so it's originally present as chromatin before mitosis occurs. And this chromatin condenses to form the chromosomes. Before exams, you just need to know these 2 points over here: condenses and becomes visible. Those are the 2 key points that are really important. In animal cells, the centrioles located in the cytoplasm near the nuclear envelope separate and move to opposite poles of the cells. So what happens if I go back to this diagram over here? If I just show you the centrioles, these are your centrioles over here. Okay, these are your centrioles. And what happens, you can see that the chromosomes are present like this. The chromosomes are present like that. Okay, and what happens during DNA during mitosis is that these X chromosomes, they are separated. So this will move to one end, this will move to one end. The 2 single chromosomes, they're separated from each other, they're pulled to either end. And that work is done by these things over here called the centrioles. Okay, these centrioles, they release spindle fibers. Okay, that's basically what happens. Okay, so the reason why we have these centrioles is so we can pull the X chromosomes apart from each other. So you have one single chromosome at each end of the cell. So basically, as you can see in anaphase, they've been pulled apart, and one chromosome is going to one end and the other chromosome is going to the other end. So if we have a look over here, we can see that in animal cells, the centrioles separate and move to opposite poles of the cell. So these centrioles, they're going to be releasing spindle fibers. Okay, and these spindle fibers, they will pull apart the chromosomes. So we have these chromosomes, one will go this way, one will go that way. Um, the spindle fibers, they are released from the centriole. So let's say this is centriole, this is a centriole. Um, this will release a spindle fiber and it will attach here. This will release a spindle fiber and it will attach here. And then it will pull one chromosome to either end. Each chromosome will be pulled to its either end. Okay, and you can see in a chromosome, we have these X chromosomes, and we can see that these X chromosomes meet at the center. The point at which they meet is called the centromere. Okay, this is the point where the 2 chromosomes are joined together. Okay, so keywords there over here, in bold, you've got centrioles which are present in animal cells, okay, they will go to either end of the cytoplasm and they will release these things called spindle fibers. And these spindle fibers are going to attach onto the centromere of the chromosome. The centromere is just the middle part of the chromosome. And they're going to pull apart the chromosome. All of this doesn't happen in prophase. This all of this just prepares itself to happen. So basically, your centrioles will move to either end of the cell. Okay, so basically, centrioles move to either end of the cell. That's basically what happens. Okay. This is some extra information. Plants have spindle apparatus but lack in centrioles. And another key important thing is that the nuclear envelope will start to disintegrate at the end of prophase. Okay, so the key bits of information that you need to take from this slide for exams, prophase, is that before mitosis, the chromosomes are invisible. So you won't be able to actually observe or see the chromosomes physically. In prophase, the chromosomes will condense and become visible. These are both always 1 mark each. They will condense and become visible. The centrioles will move to either end of the cell. So opposite ends of the cell, the centrioles will move to opposite ends of the cell, and then the nuclear envelope will start to disintegrate. That's all you need to know for prophase. So it's a long paragraph. Reason why is just I wanted to explain some of the concepts to make it more understandable. But what I've ticked over there are the key points that you need to know for exam purposes. So the chromosomes condense and become visible. The centrioles move to either end of the cell, and the nuclear envelope starts to disintegrate. Okay, so that's prophase. Now what we're going to move on to is the next stage. Okay, so remember PMAT, we've done prophase now. We're now going to move on to the next stage. Now the next stage is metaphase, M for metaphase. Okay, and this is what happens in metaphase. The chromosomes align along the equator of the plate. Again, this is really important in terms of mark schemes. The spindle fibers, which are released from the poles, now attach to the centromere and chromatid. Okay, now what that actually means is that, so we've looked at prophase, okay, we've looked at interphase already, we've looked at prophase, we can see that these chromosomes have condensed and become visible. And we can see that these centrioles with spindle fibers have moved to opposite ends of the cell. Now, in metaphase, what happens? So, if we think about the equator is literally the middle. So, the chromosomes, as you can see, they're just random at the moment. These chromosomes are just randomly placed. They're placed in like a four shaped circle there. But the chromosomes will line up in a straight line at the equator of the cell. Okay, so as you can see, the chromosomes line up in a straight line at the equator of the cell. So that, as you can see, it's a straight line. And remember when I said the centrioles release spindle fibers, so remember these centrioles over here, these are centrioles. These centrioles, they release spindle fibers. So as you can see, the centrioles are at the opposite ends of the cell. They're releasing these lines over here, as you can see. They're releasing these lines. Each one is releasing lines. Okay, like that. The centrioles are releasing spindle fibers and they are attaching to the centromere. Remember, if I draw this X chromosome, it's the middle part of the chromosome. They attach to the centromere of the chromatid, and chromatid is just, so if we draw an X like this. For example, as you can see, there are two lines. Each line is called a chromatid. So in an X, you have two lines. Each line is called a chromatid. Okay, so to summarize, in metaphase, what's happening is that the chromosomes are now lining up at the equator of the cell in a straight line. The centrioles, which have moved to the opposite ends of the cell, are going to be releasing their spindle fibers. So they're going to release their spindle fibers, and the spindle fibers are going to attach to the center of the chromosome. They're going to attach to the center of the chromosome where the two chromatids meet in the middle, and that's called the centromere. So the spindle fibers are going to attach to the centromere of the chromosome, and it's going to pull each of the chromatids to opposite ends of the cell. So that's what happens in metaphase. Okay, so moving on to anaphase. In anaphase, again, it's a bit more information than you require, but again, I'll highlight the key parts that you need to know. This is what happens in anaphase. So remember, we're at a stage where the centrioles have released their spindle fibers. The spindle fibers have attached to the center of each chromosome at the centromere. Okay, so it's just attached. In anaphase, the centrioles are going to start to retract. Okay, they're going to start to pull the spindle fibers back towards them. Now the spindle fibers have already attached to the centromere, and now the centrioles are starting to retract. So basically what they're going to do is they're going to pull the chromatids to opposite poles of the cell. So they're going to start to retract and pull the chromatids towards opposite poles. So this first point is really important. This causes the centromere to divide into two, and the individual chromatids are pulled to each opposite pole. So what happens here, if I go back to the diagram, is that the chromatids are going to be pulled to opposite ends of the pole. Okay, and this part over here is really important as well. Now, the centrioles are retracting. They are pulling the spindle fibers which are attached to the centromere. How do they pull? How do they retract? It requires energy. It requires energy provided by respiration in the mitochondria. Okay. So the key points there, the first and the last point there. What happens in anaphase, just to summarize, is that you have your centrioles, they will release your spindle fibers, and they've attached already to the centromere, but they will start to retract. They will start to pull the individual chromatids to either end of the cell. Okay, and this process will require energy because obviously this is not a ready-made process. It will require energy, and it gets this energy from ATP, which is provided in respiration from the mitochondria. Okay, so we've looked at prophase, we've looked at metaphase, we've looked at anaphase. Okay, there's only one more process that is left, and that is T, which is telophase. Okay, so first of all, if we have a look at anaphase in our diagram, so we had metaphase over here, where these centrioles have attached to the centromere of each chromosome. The spindle fibers have attached to each of the centromeres from each chromosome, as you can see. And in anaphase, the only thing that happens is that these spindle fibers will start to retract. Okay, so the spindle fibers start to retract and pull each individual chromatid. So remember, each individual one is called a chromatid. Each chromatid is pulled to opposite ends of the cell. Okay, so that's what happens in anaphase. Now in telophase, it's really short what happens in telophase. In telophase, you've got these two key points. As you can see, the chromosomes are at each end of the pole. They become longer and thinner. Key point there. Longer and thinner. The spindle fibers start to disintegrate and the nucleus starts to reform. So I've made it very concise there. That's basically what you need to know in terms of telophase. In telophase, your chromosomes have separated. So your chromatids have separated. The chromosomes which have separated now, they will become longer and thinner. Before they were shorter and fatter. At the end, they become longer and thinner. Okay, and the spindle fibers which were pulling the chromosomes, pulling the individual chromatids to either end of the pole, they will disintegrate because they need to detach from the chromatids. They don't want to stay attached to the chromatids, so they need to disintegrate. Okay, so the spindle fibers start to disintegrate. The chromatids are there by themselves now, and then the nucleus will start to reform. So as you can see over here, the nucleus has started to reform. These chromosomes have become longer and thinner. Okay, so in telophase, your nucleus is basically reforming, and these chromosomes have become longer and thinner. You've still got your centrioles there. However, there's no spindle fibers. Okay, the spindle fibers have disintegrated. Okay, so that's what happens in telophase. And as you said, as we already know, as we touched upon, cytokinesis is your last stage to form your daughter cells. The cytoplasm splits to form two genetically identical cells. So remember, in cytokinesis, it's your final stage and it's forming your identical daughter cells. It's your division of your cell membrane and your cytoplasm, and you have your two identical daughter cells which have been formed. Okay, so that is telophase and cytokinesis. Okay, so that's basically the whole stage of mitosis. We had interphase, where the chromosomes are invisible. That's where your organelles replicate, the cell grows in size. You also have your error checking stage in your interphase. That's not actually part of mitosis. Then you move into mitosis. The acronym that you need to remember is your PMAT. You've got your prophase, metaphase, anaphase, and telophase. That's your acronym. In prophase, the chromosomes will firstly condense and become visible. Key point that condense and become visible. Okay, and then your centrioles will start moving to opposite ends of the cell, and the nuclear envelope will start to disintegrate. Then you've got your metaphase. Your metaphase, your chromosomes will align at the equator of the cell in a straight line. Okay, and what will happen is that the spindle fibers, which have come from the centrioles, they will attach to the middle of each chromosome. That's where the centromere of the chromosome is. They will just attach to the centromere of the chromosome. In anaphase, the centrioles will start to retract the spindle fibers. Okay, so they will start to pull the spindle fibers in. And the spindle fibers, they're attached to the middle of the chromosome. So what they will do is that they will pull each individual chromatid towards opposite ends. Remember, when we have an X chromosome, one line is showing a chromatid. So each line is showing a chromatid. A chromosome is consisted of two chromatids. Okay, so each chromatid is pulled towards opposite ends of the cell. Okay, that's what happens in your anaphase. And then your telophase, your chromosomes, which have been pulled to either end of the cells, they will become longer and they will become thinner. And then the spindle fibers, which have attached to the chromatids, they will disintegrate because you don't want the spindle fibers attached normally. So the spindle fibers will disintegrate and the nucleus will start to reform. That's your mitosis. And then finally, you want your two identical daughter cells to be produced, and that's why you have cytokinesis. That's when your cytoplasm and your cell membrane split to form your two identical daughter cells. Okay, and in terms of content, we're just going to finish off by looking at the mitotic index. Now, the mitotic index is really simple, and in an exam, it's normally worth only 1 mark. But basically, the mitotic index is calculated by counting how many cells are visible in the field of view and the number of cells that are in mitosis. Okay, so if I think, yeah, let's say I'm looking at this area over here, and you've got these many cells. Okay, there's these many cells that I can count. So firstly, you count the number of cells. Okay, so let's say there's 12 cells over here. Okay, you then count the number of cells going through mitosis. So let's imagine I said this is going through mitosis, this is going through mitosis, and this is going through mitosis. So I said that there were 3 cells which are going through mitosis. You would then plug in these values into this formula over here. The number of cells in mitosis divided by the total number of cells that you can see multiplied by 100. Sometimes you can leave your answer as a decimal as well. Okay, so your total number of cells in this case would be 3, as in mitosis would be 3 divided by the total number of cells that you can see, which would be 12, and then you would multiply by 100, which would mean that you have 33.3%. Okay, but normally you can leave your answer as a decimal, 0.3. Okay, and that's basically mitotic index.